It is early Friday morning in Beaufort. Research vessel Eastward is coming home after a week at sea. Her mission has been to sample the sediments of the Carolina Shelf. The scientists aboard hope their collection of rocks and sediment will add useful data to their study of the ocean and its coastal areas. There is much specialized and complex gear to unload: underwater television cameras, dredges, coring pipe, and the samples collected along the continental shelf. The crew is ready for 2 days' rest, and for a change, Eastward can lie quietly at her berth. 117 feet long, displacing 74 tons, Eastward carries 15 scientists and 15 crew. She can carry enough food and fresh water to last for 3 weeks at sea. A floating laboratory, Eastward is typical of many such ships at work around the world. But she doesn't have long to rest, for over the weekend a new scientific party has come to Beaufort. And by sunrise Monday morning, the dock is again crowded with gear and people as the loading for a new cruise begins. Scientists from several laboratories have gathered for this expedition. Biologists, geologists, physical oceanographers, each interested in some aspect of the marine environment.
And you got 70 pounds of shrimp or something like that?
That was the third night.
Yeah.
I was the only, well, that hadn't been there the first 2 nights.
Finally, preparations are complete and lines are cast off. Eastward is once again setting out to sea. Slowly, Captain Sandoy moves his ship down the channel, toward the open ocean, taking great care to avoid the danger of grounding on the shallow bottom of the Sound.
In the event there should be some kind of disaster, the ship's officers would launch these.
The first duty of the scientist is to don life jackets for a fire drill. Safety is a prime consideration aboard any ship, and all new hands must learn Eastward's rules.
I'd ask that if you come out in this area in the afternoons to read or relax, that you not sit on these things because they could inflate accidentally and you'd be thrown over the side or at least hurt badly. These things. These are the light rings I told you about. There's several around the ship, so you wouldn't have any trouble getting one of those off the bulkhead to toss out to somebody in the event they should fall over.
In the engine room, Ted Baxter makes the first of many hourly checks. These engines not only drive the ship, but also provide heating, cooling, fresh water, and electrical power. And in the galley, Howard Wilson and Clyde Everett begin preparing the first of more than 50 meals and snacks they will serve during this cruise.
Barbecue, juice berry.
As Eastward moves out of the channel, picking up the rhythmic rise and fall of the first ocean swells, she turns southwest for a day's journey to a point off Charleston. From there, she will begin sailing to and from the coast along a series of lines called transects. Each transect consists of a series of stations beginning in shallow water and ending just beyond the edge of the continental shelf at a depth of about 500 meters. At each station, the scientists have scheduled a variety of activities designed to measure the basic properties of the ocean. Each investigator has also selected stations where he will collect data suited to his individual needs. The total cruise has been planned carefully to make the best use of the ship's facilities and the limited time they are available. It is a cooperative effort. Everyone is assigned a duty watch, 4 hours on and 8 hours off, around the clock. With a busy 2 weeks ahead, a little sleep now is not a bad idea. Early Tuesday morning, Eastward is approaching the first station. The center of activity now is in the wet lab, where preparations begin for the first instrument to go over the side.
Off station.
Station 11 to bridge. Depth is 20 meters. We're on our BT and Nansen cast.
This is a bathythermograph. A glass plate is inserted into it and the instrument is ready to be put in the water. As it is slowly lowered to the bottom, its mechanism of temperature and pressure sensors will scribe a line on the glass plate, showing how the water temperature varies with depth. When the instrument is back aboard, the slide is removed and the results recorded. At each station, the bathythermograph is always the first instrument used. Its readings will be used to determine how deep the next instruments must be. Next over the side is a Nansen bottle, one of the most widely used of all the oceanographic instruments. Each bottle carries a set of thermometers and has valves that can be closed to collect a water sample. The winch operator watches a depth gauge to place the bottles at the correct depth. A weight, called a messenger, is attached to each bottle before it is lowered over the side. Depending on the depth, up to 10 bottles may be placed on the line.
450.
When all the bottles are in place, a final messenger is dropped. As it hits the first bottle in the string, several things happen almost simultaneously. The bottle flips upside down, breaking the mercury columns in the thermometers and closing the valves in the bottles. Thus, a record of the temperature and a water sample at that depth can be brought back to the surface. As each bottle turns over, the messenger attached to the bottom slides down the wire and trips the next bottle, and so on down the wire to the bottom. Finally, the bottles are brought back aboard. All the work at this site is complete. The crew can prepare to move on to the next station. On the bridge, Jimmy Harker completes his entries in the logs.
And every time we stop or start or reduce speed, and the weather every hour, the course that we're on, the true, the gyro, the magnetic, and the weather conditions, the wind direction and the force, and the sea direction and condition, and the swell direction and the height, dry wet bulb temperature, the type of clouds and how many it is. Every time something goes over the site, the station number, and the date, the time, the latitude and longitude where it went over, the depth, and the date and the time and the position and the depth where it came up at.
The entries which the ship's officers put in the logs are often used later by the scientists to reconstruct the conditions at each station. The ship's log is thus both a legal and a scientific record of each cruise.
Bridge, this is Station 1. What's our present course, please?
About 275 at 8 knots. Be at the next station about an hour and 10 minutes.
Thank you.
As Eastward proceeds toward the next station, work continues in the wet lab. Several samples will be taken from each Nansen bottle, which will then be emptied and readied for the next station. The samples will be stored below for later analysis. Some will be frozen and taken ashore at the end of the cruise for further study. The amount of oxygen dissolved in seawater is of prime importance to marine life. A solution is added to one sample to capture this dissolved oxygen for later analysis.
Okay, 10282,
The thermometers on each bottle are read and the data recorded.
28.04, 11698 is 28.00.
Back on the bridge, Jimmy Harker is checking the ship's position. Eastward uses the Loran system for most navigation.
These are receivers for signals transmitted from different Loran stations down the coast. We're getting signals now from one at Charleston and one at Cape Hatteras. And on the chart, there's a lot of crisscross lines. This is from one station, 386, 3L1, this one is. And they're divided into 50 microseconds each, and you just interpolate between them. This is 3381, there's 3350, and there's 3400, and 3381 is right there. You just interpolate between them, and you get a line of position one way. Then you switch it over on another channel. We're using 386. You set it up. And that's 4136, and you have another group of lines crisscrossing that, and wherever they cross at, that's where you are. You just triangulate them together. And of course at 10 to 4 we were right here. We're about 2.5 miles away, and we're making about 8 knots. So that's every 30 minutes we go 4 knots. And so you can just figure your time from there. That's probably the greatest help to navigation today is Loran. Is simple, and it's easy to use, and it's real accurate. These sets sometimes are even as anywhere from a couple hundred feet to right on it, depending on the weather conditions and the time of day. Nights, they're not quite as good as they are in the daytime.
Eastward's officers often use the signals from a satellite fed into a small computer on board. This gives them a check on their latitude, longitude, course, and speed.
They almost changed course to 282. Harold, somebody.
The accurate navigation of eastward is vital, for without knowledge of the ship's exact position, the information gathered at each station would be useless. As the hours pass, watches change, bringing a new group to take the samples, read the instruments, and run the ship. The ship settles into a routine, working, eating, and sleeping, all governed by the observations at the next station or the experiment underway. The ringing of Clyde Everett's bell signals time to eat. From early morning until after midnight, Howard Wilson's galley is alive with taste-tempting odors. Eating is a favorite activity on Eastward, and good food is a tradition. By Wednesday night, enough stations have been visited for the first batch of Nansen bottle samples to be taken to the dry lab below decks for analysis. At each station, the bottles of water taken for oxygen analysis were treated to capture the dissolved oxygen in a chemical compound. Now the treated water in each bottle will be tested in turn and the amount of dissolved oxygen determined. John Croyder is performing another test on a sample of seawater.
This machine simply measures the conductivity of seawater. Gives us a measure of the amount of salt in any given sample. First we balance the temperature, then we switch it over to salinity. .67879. From this we take the conductivity reading, look it up in a table for the appropriate temperature, and calculate the salinity.
Salt content, oxygen content, temperature, and depth, currents, wind, and weather. All taken together, these factors plotted station by station can lead to a picture of the water of this coastline. The scientists want to know what these waters are like. They want to establish a base of information that can serve as a background for future studies. Putting a message in a bottle and throwing it overboard can still be useful. Bottles like these are cast adrift at several stations close to shore. When found by people on the beach, the cards inside these bottles will be filled out with the time and place where the bottle was found. This data will help create a picture of the surface currents near shore. This station is also one of those selected to study another aspect of the marine environment. Terry Lynn Herbert and Evelyn Haynes are studying primary productivity. Terry begins each time by lowering a device called a Sankey disc over the side. Evelyn describes the experimental procedure.
The term primary production is used by ecologists to mean the amount of production that goes on in transforming inorganic carbon to organic carbon. It's also called the first link in the food chain. Is the green plants. In terrestrial environments it would be grass or corn, and plants can only produce in the area of the ocean in which there's enough light for them to use as energy to transform the inorganic carbon in the water to organic carbon of living tissues.
The white Sankey disk, reflecting light back to the surface, is lowered until it can no longer be seen.
No. Can you see? Wait a minute, wait a minute.
The depth where the disk disappears is noted for future reference.
Okay, what you got? Okay, and bring her up.
By taking a Secchi disk reading, you get an idea of the amount or the extent to which light is attenuated in the sea. When you get perfectly clear ocean waters, light goes down to maybe 100 meters till it's absolutely dark. In inshore waters, light may go down a couple meters or 5 meters until it's absolutely dark.
Next, Niskin bottles are used to collect water samples.
And we take the water from various depths that have been worked out according to the light attenuation measured by the Secchi disk. And we take them at points in the water, depths in the water at which there's 100% of the instant light radiation on the surface, which is right at the surface.
62.
At 50% of the light that's shining on the surface, at 25%, at 10%, and 1% of the instant light of the surface.
A second bottle is added, and then a third. Usually 4 or 5 bottles are placed at various depths on the line. Again, messengers are dropped down the line to close the valves on the bottles. Then they are brought back on board the ship.
So what we do is put radioactive carbonate, inorganic carbon, in the water in those little bottles. And what Terry Lynn is doing is measuring the amount of inorganic carbon that plants, the little one-cell plants in the water, take up in a 24-hour period. And we do this all across the shelf for, these 6 transects. And we get an idea of the total primary production that the shelf waters are capable of. And this primary production is where everything else in the shelf waters gets its energy, its food, all the fish, shrimp, everything else. Chlorophyll is another indication of the amount of primary production that can go on in a particular spot. And since chlorophyll is the organic molecule that makes a plant capable of using sun's energy to convert inorganic carbon to organic carbon, the more chlorophyll you have, the more production that can go on, although that doesn't necessarily mean that production is going on. So we filter a certain amount of water, usually it's about 100 milliliters to 300 milliliters of water. It's then on a very small glass fiber filter pad. We take it down to the laboratory, grind it up in the tissue homogenizer with the electric drill until it's all ground up. And this is ground up in 90% acetone, which is organic solvent with a little bit of water mixed in it. The chlorophyll A that was in the phytoplankton cells then is extracted into the acetone, what you have is molecules of chlorophyll A. After we grind it up, we put it into the centrifuge to spin down the remnants of the filter. All the chlorophyll A is in the acetone. Then we analyze it in an instrument which uses UV light. Chlorophyll A fluoresces. So if you shine UV light into the chlorophyll A, it will produce a light of its own, and this is picked up by a sensitive photocell. This machine is really very sensitive to any small amount of chlorophyll A in the water.
While Evelyn and Terry Lynn are studying microscopic plant life, John Hall is studying microscopic animals called zooplankton, one step higher on the food chain.
I'm interested in two things, basically, working with this zooplankton. One, I'm interested in trying to answer the question, could the shelf zooplankton be an important source of nitrogen, inorganic or organic forms that could be used by phytoplankton? Could they be important in regenerating this nitrogen fast enough so that they could play some significant role in controlling phytoplankton productivity? And so my basic thing is saying, okay, well look, you've got zooplankton feeding on these phytoplankton, so they're obviously taking the nitrogen that the phytoplankton accumulate, they're taking it into their own bodies and they're doing something with it. Eventually they're recycling it, and they're changing it once again to a form of nitrogen that the phytoplankton can use, and I'm just interested in how fast they're doing this. Do they do it in 5 days, or is it going to take a month, or is it going to take 6 months? That's one half of the thing. The other half of the thing is I'm interested in the respiration of the animals, because a combination of the respiration information and the nitrogen excretion information gives you some indication of whether the animals are feeding, or whether the animals are starving, or whether they're halfway in between. And so those are the two things that I'm interested in doing.
And so Eastward steams on. Slowly, a pattern is emerging in the data. The character of the water beneath the ship reveals an intrusion of cold, nutrient-rich water from the deep ocean onto the continental shelf. The data from studies of primary productivity, respiration, and more show a healthy marine population, only beginning to feel the effects of pollution from sources on land. But there are still unanswered questions. What are the pollution levels? Is the level of air pollution related to pollutants in the water? How does the intrusion of water masses affect the higher forms of marine life? The cruise is far from finished, and so Eastward sails on.